Hi, good day ma'am. So we are the group 3 and we are going to discuss the exercise 11 entitled Population Genetics, the Hardy-Weinberg equation. So first, we have our observation. We have survey 100 students in CMU of normal fifth finger and hitchhiker fifth finger. We have gathered the data of normal fifth finger of 82 and 18 in hitchhiker fifth finger.
In table 21, the frequency of big B, which is the normal finger, is 82, and the frequency of small b, which is the bent finger, is 18. So first, we need to find the allelic frequency by the formula P plus Q equals 1. So, dili man na to una makuha ang P, so unahon na to ang Q. So, Q squared equals to 18 over 100. Then Q squared equals 0.18. Then we need to square root both sides to find the Q, which is the 0.42. To find the P, we need to minus 1 minus the answer of Q, which is 1 minus 0.42, and the answer is P equals to 0.58. After finding the allelic frequency of P and Q, we can now find the genotypic frequency with the formula P squared plus 2PQ plus Q squared equals 1. The P squared here is the homozygous dominant, the 2PQ is heterozygous, and Q squared is the homozygous recessive. So, 0.58 squared equals to 0.34. 2 times 0.58 times 0.42 is equals to 0.49. Then 0.42 squared is 0.18. So if we add that all, the answer is 1. In table 22, we just substituted the values in table 21 in the Punnett square.
Table 23, Algillus and phenogenic sequence of generation number 2, the given P sequence of big B in normal fifth finger is 75 and the Q sequence of small b bend fifth finger 25. Phenotype normal fifth finger, bend fifth finger, and the genotype is first is double big B, second big B small b, and third two double small b. And the genotype sequence, the P squared is equal to 0.25 and the 2PQ is equal to 0.5 and Q squared is equal to 0.25 and the gamete sequence is equal to 0.5 and the other one is 0.5. For table 24, alleles and the phenotype sequence of generation number 3, the given P sequence of big B normal fifth finger is 76. Q sequence of small b bent fifth finger is 24. Phenotype normal fifth finger, second normal fifth finger, third bent fifth finger. The genotype is doubles big B, second big B, small b, third double small b. The genotype sequence, the P squared is equal to 0.16, 2PQ is equal to 0.50, Q squared is equal to 0.24. The gamete sequence is equal to 0.51 and lastly 0.49.
Table 25 shows the individual and class observation for some human traits. What are these human traits? Number 1 is the widow's peak. It has an allele of a big letter W. Its phenotype is normal peak. Genotype is heterozygous dominant W, and its total observation class is 17. The second one is the attached earlobe. Its allele is a small letter A. Its phenotype is free earlobe. Its genotype is homozygous dominant AA, and its total class observation is 20. Third one is the Darwin's ear point. Its allele is big letter D. Phenotype is Darwin's ear point, and its genotype is homozygous DD, and its total class observation is 23. The fourth one is the cliff chin. It has an allele of a big letter C. The phenotype is no cliff chin. Its genotype is homozygous dominant C. The total observation per class is 7. The fifth one is the tongue rolling. Its allele is a big letter R. Its phenotype is normal tongue. Its genotype is heterozygous RR, and its total class observation is 68. The sixth one is the tongue folding. Its allele is a big letter F. Phenotype is no normal tongue. Its genotype is homozygous homozygous dominant FF, and its total class observation is 24. The seventh one is the hitchhiker's thumb. Its allele is a big letter H. Its phenotype is hitchhiker's thumb. The genotype is homozygous dominant HH, and its class observation is 21. The last one is the dimple. Its allele is a big letter D. Its phenotype is dimple, and its genotype is heterozygous dominant DD, and the total class observation is 49.
In order for us to answer table number 27, it was included in the procedure number 3, which is the effect of small population and the allele frequency, where we need to obtain a total of 70 red, 30 yellow, and 20 blue buttons. In this activity, we didn't have a buttons, and we used an alternative in order for us to get the combination of generation 1. For the value of P, we just need to do the same process as getting the value of Q. For example, the formula of P is P is equals P squared plus 1/2 times 2P Q, and the genetic frequency, which will always equal to 1. In continuation to our table number 25, table 26 is where we put our answers for the equivalent of PQ and 2PQ. In this table, we computed the genetic frequency of the following traits, which are the widow's peak, attached earlobe, Darwin's ear point, cliff chin, tongue rolling, tongue folding, hitchhiker's thumb, and the dimple. In this table, there are a total of 10 generations, and each generation is composed of 10 individuals, where we use 10 as the population in order to compute the P, Q, and the 2PQ. In this table, table number 27, our big RR is the red, the small RR is the blue, and the big R, small r is the yellow. Big RR is our P squared, the small RR is our Q squared, and the big R, small r is our 2P squared. For example, in generation 1, we get a total tally of 118. We got 8 red, 1 yellow, and 1 blue, with a total of 10 population. In order to get the Q squared, we need to divide 1 over the population, which is 10. This will result to 0.1. And in order for us to get the value of Q, we just need to follow the formula Q is equals to Q squared plus 1/2 times 2PQ.
In conclusion, to conclude the Weinberg equation, the amount of genetics variation in population will remain constant for one generation to the next absence of distributing factor. In part 7, in the study guide question, in the first part here, the state the Hardy-Weinberg equilibrium, it says that can be distributed by a number of forces including mutation, natural selection, non-random mating, genetic drift, gene flow, genotype frequencies in a population remain constant between generation. Number 2, what condition should be satisfied in order for the original proportion for the alleles in the population remain uncharged from generation? The answer is no charge in DNA sequences, no migration, a very large population size, random mating, and no natural selection. Number 3, give application of Hardy-Weinberg equilibrium. The answer is to predict genotype and alleles frequencies in the future generation, assesses the equilibrium and of current population and interpret the genotype. Number 4, what is the genetic drift? Which part of the exercises demonstrated this concept? It is change frequencies of an existing gene variance in a population due to random chances. So this exercise number 11. Number 5, what is the founder effect? Which part of the exercise demonstrates this concept? Loss of genetics variation that occurs when a new population is established by a very small numbers of individuals from a larger population.